One thing you'll discover when you start looking at phylogenies is that there's a lot of terminology. In some ways, it's almost like learning a second language. I guess science in general is like that. So I'm going to try and emphasize those. Some of them are similar to one another. I will emphasize that, make sure you're clear about the difference. But there are some more vocabulary words that we need to look at. In the readings for these last couple lectures, we've been looking at why phylogeny is important. Phylogeny in many ways gives us a roadmap, a foundation, if you will, for understanding why things are different or why things are the same. And it's critical that we have that phylogeny. So for instance, if we see two eggs, maybe we see a light blue one here and a light blue one there from different species, but they have this feature that is very, very similar. It is an incredibly important question for biologists as to whether or not that similarity arose because they inherited it from a common ancestor, or if they have a similarity because it arose independently two different times. And to do that, we need a phylogeny. So if those two species are very closely related to one another, in fact, if they're their own sister taxon, then the best explanation for why they share that similar characteristic is because they inherited it from a common ancestor. However, if they're very, very distantly related and all of the intervening lineages have different features, different colored eggs in this example, then it's clear that that feature arose independently. And that's exciting and interesting. Then biologists can study the mechanisms of evolution, maybe even the physiological mechanisms that lead to the characteristic that they're looking at. Okay? In addition to just really helping us to have a roadmap for species, it also has many other alternative uses. So for instance, if we have samples of viruses, for instance, and we're able to determine how those viruses are connected to one another, then we can have a lot of information about epidemiology, about outbreaks, about how that virus evolves and changes in response to immune systems or in response to medical treatments. You know, maybe we've got an antiviral that we're giving it. We can look at mutation rate and how the virus responds when exposed to an antiviral. So, and that's really just the tip of the iceberg. There are many, many other practical and useful things that biology can do with phylogenies. In fact, every single branch of biology relies very, very heavily, some more than others, but they rely on phylogeny. So you really can't underestimate the importance of phylogenetic inference and having a good, well-supported phylogeny for the organisms that we're looking at. So here is where the vocabulary begins. We're going to begin with a rather simple idea. Analogy, if we're going to be very broad, is simply similarity. But for this purpose, we're going to look at a specific type of similarity. So analogy is similarity that is gained independently. So two things are analogous if they have a similar feature, but they didn't arise at that feature together. So for instance, let's say that you were writing an essay and your classmate turned in an essay and they were similar, but you had never looked at each other, you didn't cheat, you didn't copy the same material, you just had similar ideas, right? Which happens a lot. So those would be analogous ideas. The other concept here, which is also very important, is homology. And homology is similarity, but it's similarity that is due to inheritance from a common ancestor. So if you and your neighbor, your friend, your classmate had both used the same source material, or, you know, hopefully you don't do this, but if you had both cheated and used each other, you know, copied each other's material, then that would be homology and you would be in trouble for it. But in nature, when we see homology, we know that those organisms had a common ancestor that gained the characteristic and then they passed it down to other features. So let's look at a very simple two different hypotheses. These are unrooted trees, but they say different things. So the green lines here are connecting four species. Again, where they come together, even though we don't have a root, but where they come together, that represents a common ancestor. And so this one says that the bat and the seal are most closely related to one another, and there's their common ancestor. This one says the penguin and the eagle are most closely related to one another. There's their common ancestor, and then somewhere they connect together. And if we had an outgroup, we could root this tree, it would be somewhere in here, and then we could add other species. But for now, we're only looking at these 4. Okay, here's an alternative hypothesis. This one says no, the penguin and the seal are closely related to one another, common ancestor there. Bat and eagle are closely related to one another, there's their common ancestor. So, which of these hypotheses is better? Now, when I ask this in a live class, typically what students will say is, oh, this one up on the left is better, and then, of course, this is why you don't answer questions in class, right? I ask why. Explain it to me. And most of the time, the answer is, well, these are both birds, and those are both mammals. And that's actually quite a good answer, because if we're doing everything correctly, those taxonomic groups, mammalia and aves or birds, right? Those are based on these shared characteristics. They're based on these phylogenies. So it's actually not bad. But I will then say, okay, great, they're mammals, but what does that mean? Why do we group them together in mammals? What are the shared characteristics, right? Or the homologies that identify these as mammals? Now, if you looked at them and said, oh, they have a backbone, that's not a good question. So we're not only looking, those are homologous characters for a bat and a seal, but it doesn't help us with this problem, right? Because penguins have backbones and eagles have backbones and many other non-mammals and non-birds have backbones, right? So not only are we looking for a homology, but we're looking for a homology that unites and is unique to the group that we're talking about. And we'll talk about that term here in just a little bit, okay? So many groups are named for these homologies that unite them. So mammals are named after the mammary glands, right? These are these structures that allow females to feed their young. There are other mammal characteristics also, though. So we could look at hair, right? A follicle. And some mammals have more than others. Some covers their whole body, some just partial. But all mammals have follicles. And there are other mammal characters that if you knew the group well, you would be fine. And we already talked about that with birds. And you remember we even introduced the term with birds. It's a synapomorphy. So a synapomorphy, right, which I've got here on the next slide, is a homologous character, a shared ancestral characteristic that everybody in the group still has. Because if some of them have lost it, that's called a simplesiomorphy. We'll have that up on the screen in a bit. If some organisms have lost it, then it no longer serves as a single uniting feature, okay? And that can be a little bit tricky, right? So this hypothesis is better, not only because these are mammals and these are birds, which is all right. If our classifications match our phylogenies, that works just great. But also you can say because these guys share many more synapomorphies, despite having some differences, than they do with other organisms, right? And these guys share more synapomorphies. So if I say, oh, the bat and the eagle can fly, there's a potential that that's not, in fact, in this case, it's not, right, a synapomorphy. In fact, it's not even homologous. Those features arose independently, so they are a form of analogy. And how do we know that? Well, we know it because when we look at a phylogeny, mammals are only very distantly related to the birds. And even though a bat and an eagle may share some superficial similarities, when you start looking at all of their characteristics, it's very clearly apparent that they're not as closely related to one another as the eagle is to the penguin or as the bat is to the seal. Okay? And we're actually going to look at a method, and we'll outline it today and on the worksheet, which I'll give you a little bit of pointers on the next lecture. Gives you an exercise to use that method to determine which of a 2 or 3, we'll only do 2 on the worksheet, but you could do dozens of different phylogenetic hypotheses, which is the best. Okay, so again, a synapomorphy is any ancestral characteristic that all of the descendants still have, and many groups are named after synapomorphies. So feathers are a great synapomorphy for birds. Now flight, right, is not a synapomorphy for birds for actually a couple of reasons. If we're just going to say they can fly. Number one, there are other things outside of the birds that also can fly. So it's very obvious that those instances of flight evolve separately. Number two, some birds, penguin included, have lost the ability to fly. So that no longer is a synapomorphy, a unique defining characteristic for the group Aves. Okay? Now, one method that is used to help us find the very best phylogeny is parsimony. And parsimony is simply an exercise where we try to find the simplest hypothesis. This actually goes way back to a philosopher from the Middle Ages called Occam. And so sometimes parsimony or related concepts are referred to as Occam's razor, because his idea was that, although it's not always correct, that the simplest explanation is most often the correct one. Okay? And I use this analogy in class. I'll talk about it now. This is a good kind of introduction to the idea of parsimony if you're not familiar with it. Let's say you own a dog, and your dog is an inside dog. You leave it at home while you go to school or work, and you come home one day, and one of the pillows from your couch is all ripped to shreds. It's a little bit damp and wet, and your dog is sitting in the corner with its tail between its legs. That's the data you have. That's the evidence. What happened while you were out? Well, almost certainly you would go to the simplest explanation, that your dog, even if it had never done this before and didn't have a history of it, your dog ripped up the pillow, and it's kind of wet because it's got a saliva on it. You can maybe get more detailed, but that's the simplest explanation. Now, is it possible that your dog was framed? Right? Maybe the FBI broke into your home, and I don't know why they would do this, but they ripped up your pillow, they put fake dog saliva on it. Your poor dog is traumatized in the corner, scared, as some dogs will be, because they had intruders in the home. Is that possible? And it doesn't break any of the rules of our universe as we know it. So yeah, that's possible, but not very likely. So in parsimony, we're doing a similar thing using characteristics. They can be morphological, they can be genetic, and we're using characteristics to find the simplest explanation. And the reason why we need to do that is some characteristics might contradict other characteristics. So for instance, let's say I think flight is a good character. And so I have to put flight on this phylogeny. If this is my best phylogeny, I would put flight having evolved twice. And to do that, we mark lines. In fact, let me go ahead and I'll put it on here. We mark lines across branches. And so we would put a line here, right? We'll move it so it's there across the branch. And then we put an identical line across this branch. And we'll do this on the worksheet on the exercise. And so those lines represent evolutionary events. Now notice we've had to put a line on there twice. So this is not a very simple explanation, right? We prefer simple things where things happen once, but that's not always going to be the case. But if flight was my only characteristic, if that's all that I was using, then what I would do is on this tree, I could mark a line once, come back here. Let's do the same thing. I could put a line once across here representing, and it doesn't matter where, as long as I get it on the correct line. It doesn't matter if I do it on this side or in the middle, but somewhere along this line, I would have the origin of flight. And only have to do it once. That's a very simple explanation. So if flight is the only character we're using, this phylogeny is not as good as this one because this one's simpler. I only have to infer one origin. I would say flight in bats and eagles is homologous, and I would be done. Now, we don't do that. We don't use just one characteristic because sometimes characteristics can be misleading. They could have evolved twice, or it could have evolved once in a very ancient ancestors, and then some of the descendants lost the ability to fly. So what I do is I put together a large data set. We sometimes call it a matrix. And this data set is all of the characteristics, some of which are going to be synapomorphies. Others may have evolved convergently, like flight in the bat and the eagle, right? So convergence is a good term. We'll get to it here in a little bit more detail later. And so the idea is of parsimony is that if I gather all the characters, I treat them all equally, then overall the simpler ones are going to be more common, and so they'll help me to understand relationships better. Okay? And so even though I have to map one character on here twice, if I did other characteristics, and let's do it one more time. I'm going to pop out of here one more time. So let's say, and we'll even maybe make them different colors here. So if I make this character, I'll probably do the outline. I put that character on there. Maybe that character represents the origin of the mammary glands, right, which separates the mammals from the birds. I only have to do that once. And then if I put on here in a different one, the origin of the feather. And let's make that one even another shape, just so we can kind of tell them apart. So the origin of the feather. And again, it really doesn't matter which way I put them on there. If we had a rooted tree, then where I put them, if there's more than one characteristic, might make a slight difference. But for the purpose of just determining which phylogeny is better, it really doesn't matter. Okay? So now I have two characters that say no. You know, it's simpler. We can map them on there just once if the mammals are closely related, if the birds are closely related. And so this is now a best explanation because I only have to map those characters on there once, and I have to map the other character on there twice. But this is simpler. And we're going to look at that in the next lecture. I'll do a little bit at the end of the worksheet problem to help you, give you some pointers with that. Now, here is a term that is confusing for some students because it's so similar to homology. And even though it looks similar, has the same prefix, Greek prefix meaning the same, it's not the same. It's not the same word, and you need to make sure you don't confuse them. Homoplasy is when we have to have a more complex explanation. So homology is simply similarity due to a common ancestry. A synapomorphy is a form of homology that is the simplest kind, where it just happens once and then everyone still has it. Okay? But homoplasy, right, occurs when we have a more complex explanation. And there are two forms of homoplasy that we will see as we're doing our exercises. The first we've already mentioned is convergence, right? Convergence is where a similar characteristic evolves independently in two separate lineages. So once we know that this is the relationship, it's very clear that flight is a convergent characteristic. It evolved once in bats and once in birds. Now, the easiest way, the best way to determine if something is a convergence is we map it, like I was doing with those lines, onto a well-supported phylogeny. We mark the lines onto a phylogeny. It's called mapping characters. And if they occur separately in two different lineages, it's convergence. Often, even before we had very well-supported phylogenies, we could tell that things were convergent. So you can get a pretty good idea about convergence by looking at the very fine features of a structure also. Bird wings and bat wings, although superficially similar, once you look at all those fine features like the skin for the bat wings stretched between fingers, the large feathers of the birds and the reduced bones, it's very clearly that they're not all that similar in the fine details. And so that's a good indication, not the best way to do it, but that's a good indication that they are convergent. Okay, so the best way is to map them onto a well-supported phylogeny. All right, our next form of homology is simplesiomorphy. Now, simplesiomorphy is where we have an ancestral characteristic, so it's a homology, but some of the descendants lose those ancestral characteristics. Okay, and that's simplesiomorphy. So for instance, if I marked the origin of flight in birds, it actually didn't occur in the eagle, and then the penguin never evolved flights. Penguins had flying ancestors. All birds had ancestors that could fly at one point. However, the penguin has lost that. And if we added more species to this phylogeny, that would be easily apparent because penguins are somewhat closely related to flying lineages. And it's very clear that they lost the ability to fly. And so we would mark the origin of bird flight actually down here, and then a second line representing the loss, representing an independent evolutionary event where we lost it. And again, you'll get practice with that in the next on the worksheet. Okay, so just know the definition, and then we'll have some practice with it. And so what we do again with parsimony is we take all of our characteristics that we think might be useful, we organize them into a matrix, and then we map them onto whatever hypothesis. And we can do every single possible phylogeny. We could do two that we think are both equally good, and we just need to test with our characters. But once we map them onto a phylogeny, we just count up all the evolutionary events, and the one with the least number of evolutionary events, the simplest explanation, is the one that we use. Okay? So here's a phylogeny. Now, notice that we have two different illustrations here. So we have convergence, where two organisms have independently lost a feature. And then we have symplecomorphy here, where we could say, oh, look, 4 and 5 are both blue, but they are not each other's sister taxon. 4 is more closely related to 3 than it is to 5. And so we have to come up with an explanation for why they are both blue. And so we can either infer that, the ancestors were light blue and then they convergently gained this blue color, right? They gained it separately. And so sometimes defining whether a character is a loss or a gain, if something's a color, are we losing something if it changes to another color? Well, really, we're just doing changes. So sometimes you really just think of losses and gains just as changes, and that's fine. But we're looking at the same character, and we look at that character with all of the changes that occurred in that character, and then we move on to the next character. So this color in this phylogeny represents a symplecomorphy. It was gained in the ancestor, then lost in one of the descendants. Now, I'm going to leave this an open question for now, but we'll come back to it in the next lecture. So parsimony treats all changes the same. And when we map a change onto a phylogeny, that's a cost. And so tree length or tree cost is adding up all of the changes that we need to infer occurred to help us explain why characters occur in different organisms. So here we've explained it with a gain in an ancestor and then a loss in one of the descendants. Okay, so this is looking at, I think, this the blue character. So this dark blue color was gained here, but then it lost. In fact, it reverted back to 3 to the light blue. Gained here and lost. So that's a complex explanation. That would be a symplecomorphy. So in this group, the dark blue color is homologous because it was gained in an ancestor, but it's also a symplecomorphy because one of the descendants lost it. Okay. Hopefully this is starting to make sense. You're going to get more practice on it, so if it's still a little bit complex, that's fine. And so we've got here our phylogeny they've got boxes. You can look at these, pause the lecture if you want to, and look at the explanation for why we have to mark things on there multiple times. We didn't have to do it with the purple one. We could just say, hey, it happened one time. It was a loss, or you could look at it as a gain of the lighter color, but a change there. That's a simple one. That's a nice explanation. But the others are much more complex. Okay, so there are two mechanisms. Remember, convergence and symplecomorphy, or loss of a feature in one of the descendants or more, that can help us explain homoplasy. Now, it turns out that with this phylogeny, there's a better phylogeny that's simpler than this one. So notice if I actually move 3 so it's more closely related to 2, and I move 4 and 5 so they're closely related to one another, I end up with a simpler explanation. So instead of having to map characters on there 1, 2, 3, 4, 5 times, I can map a character on there just 3 times. And this now means that C and B, if 4 and 5 are closely related to one another, C and B are homologous, and they're synapomorphies for this group. So they were gained in a common ancestor, and all of the descendants still have that characteristic. And so if this is our data set, if we only have 3 characters, this is a much better explanation, a much better hypothesis of relationships than this one is, because it's simpler, and that's parsimony. Okay? Now, we'll do this quickly just to give you a kind of another example of why roots are important, because we've already talked about this. To root a unrooted phylogeny, so if I have 4 species that I think are close related to one another, I want a rooted phylogeny, I need an outgroup. And I would pick something that would be similar. So here I've got a bird that's not part of the in-group, but is similar. Now, hopefully, if you're thinking about this, you realize there's a little bit of a catch-22 here. And that catch-22 is, well, I want to figure out relationships of organisms. And to get a nice root for that, I need to pick an organism that's outside of the group. So in a way, you're kind of already making a prejudgment about what organisms are related to what, at least in terms to the in-group, right, the group that you're studying, and the out-group. You're saying the out-group is not part of this group. And so that can be a little bit problematic. And so to get around that, we kind of do multiple iterations. You can pick multiple out-groups, different analyses. And it is a little bit of a problem at the outset, but It's not too much. It's something that we can deal with. And the reason we want rooted trees is they tell us something about the order of events. So in this one, if I map characters onto these branches, I can do it and get one phylogeny that's better than the other. But I don't know which characters happened first. This, however, tells me that this is a more ancient ancestor than that one. And so if I have a character mapped on that branch versus that branch, I know this one happened earlier in the history of the organisms. And we could even use it to help us understand characters that look at distribution, for instance. So where do I find organisms? So if the black and yellow-billed magpie are North American in distribution, I know that the group originated in Asia and then migrated into North America. If, however, when I did the root came out on one of these branches, right, in the ones that live in North America, then these would be more recent events, and the group would have originated in North America and migrated out and expanded into Asia. So that's one reason why roots are important. Now, when we look at both relationships and geography, where we find organisms, we call that phylogeography. Sometimes you'll see it also as biogeography, but phylogeography is a little bit better term. That's the one that we will use. So just remember the definition for that. We'll see examples of it here and there in the coming lectures. Phylogeography is using relationships among organisms to help us understand why we find them in different areas. So understanding their distribution and their geography.